How do I interpret them? All right. There we go. Thank you. And then even once we have all these data, how do we access them and use them, and synthesize them to understand cellular and tissue functions in different physiological states and disease, so we can do what we want to do, which is predict, prevent, and treat disease. So today, I'm going to tell you a little bit about the International Federation of Musculoskeletal Research Societies and a project that the Big Data Working Group is working on, which is a musculoskeletal genomics portal as a starting point for making these data accessible. Dr. Wu from Washington University will discuss single-cell RNA sequencing and how that can reveal and define cell heterogeneity and trajectories within a tissue. And then Dr. Cappellini from Harvard University will describe how to use ATAC-seq to identify open chromatin regions during cartilage development and disease. And finally, Dr. Butani from Stanford University will discuss a proteomics technique called CyTOF, or single-cell mass cytometry, and how that can be used to identify different cells within healthy or diseased skeletal tissues. So the IFMRS is the International Federation of Musculoskeletal Research Societies. There are more than a dozen societies as part of the federation right now, including the ORS. This federation was formed to open channels of communication among musculoskeletal research societies around the world. The mission is to advance musculoskeletal research globally to prevent and treat musculoskeletal diseases. The Federation is active in a number of areas. We are not competing with societies, the activities of individual societies, but we're focusing on raising global awareness, providing shared resources and opportunities to share resources, looking at collaborative projects to promote education and to present research, and a big data inventory. There's also a group called Hubble for young investigators and new investigators. It's led by new investigators. It's an online website where they can share knowledge, protocols, and other resources. So under the Big Data Initiative, I'm pleased to be pleased to be the ORS representative of a Big Data Working Group, which is shown on the right side of the slide there. It's led by Doug Keel and Amanda Sherwood. Sherwood is the outgoing director of the International Federation of Musculoskeletal Research Societies, and Federico Mascaguri is the new executive director. Federico, are you here? Can you stand? So I don't have a picture of you up here, so he's right there. So if you want to learn more about the IFMRS, talk to Federico. But under Doug's leadership, we've made a connection with the Broad Institute, and specifically the Knowledge Portal team at the Broad Institute, and Dr. Noel Burt, who's on the top of that pyramid on the left side, is our main collaborator through this. So we started with the simple idea of making genetic and related genomic data more broadly accessible and useful, and so that it could have an impact on our ability to understand and treat human disease. So we're calling this the Musculoskeletal Genomics Knowledge Portal, and it's a research and software platform to organize human genetic and related data, and to present these analyses and information to non-experts, or even experts who have difficulty identifying where these data are. So this knowledge portal takes large-scale human genetic data sets from the community. Right now we are focused on human data sets. The Big Data Working Group culls the options. Typically we're looking at published reports at this point, and publicly available data sets. The Broad team harmonizes, does quality control on the data. We also have GWAS data from other relevant large-scale consortia and biobanks, and disease-agnostic resources like 1000 Genomes Project. So I'm going to just show you three ways that you can use this portal. So so if you're interested in genome-wide association results for your favorite trait, you could use that. So you could go explore by phenotype. You see the tab that's highlighted there, and what's written in the screenshot is bone fracture. But there are many other phenotypes that you can choose from, including estimated bone mineral density. We are working on adding osteoarthritis data this portal, and that should be done in a few weeks. So if we look at estimated bone mineral density, you can pull up an interactive Manhattan plot. Again, this is a screenshot, but theoretically, if you rolled over one of those red dots, you would see a gene of interest that's associated with changes in bone mineral density. And then you can dive deeper into the portal and get a list of the genes, the actual SNP, and the p-values for which they're associated with that phenotype. The second way is you can search by a variant. So if you know a variant, you can type that into the search engine. You'll get a summary of how that variant, what's known about that variant. So you can see a number of options here. If you go to the first one, the associations at a glance, we would see that there's a high association of this variant with lumbar spine bone mineral density, as well as estimated bone mineral density. That's an ultrasound of a heel ultrasound scan, but no association with the other phenotypes that are listed in the portal, including glycemic indices and lipids. And thirdly, I think this is my favorite way, what I use it most often for, is to search by a gene. So one of my favorite genes is RUNX2, and what will come up when you search for the gene is a description of it, but you'll also see that stop sign, those 3 dots in the upper left-hand corner. If the green is highlighted, that indicates a strong evidence for an association of that gene with a particular phenotype. So in the bottom half, you can see that estimated bone mineral density and height are green. So there's a strong association that there's some variant of RUNX2 associated with those phenotypes. Yellow there's some suggestive evidence, but it's not the p-values are not as strong. And then the gray areas, there's no association statistically with those phenotypes in RUNX2. So you can, again, dig deeper, learn more about this particular variants that are strongly associated with it, where they are they in an intron, what's the p-value, and then on the far right, you can see that you can dig deeper into the data sets as well. So you can get a link to the published article, the published paper, and all the published paper and all the details regarding that variant. We're also working on adding more tracks, and one of the challenges for our field is that musculoskeletal tissues are not well represented in databases like GTEx, and so we don't have a lot of data to compare our work to, but you can see, for example, the skeletal muscle, the second track on the bottom, there's ATAC-seq reads available for skeletal muscle, and so you can align these variants that you see with different epigenomic data sets. So that's the hope. We have a lot of work to do to get to what we all want it to be, but this is our first step. And so I think the other thing that's really interesting and exciting about this is that the musculoskeletal knowledge portal is just one of many right now. There's also a type 2 diabetes knowledge portal. There's a cardiovascular disease portal. And all of these data sets are going to be integrated, so you can see if your gene of interest or variant of interest is associated with multiple conditions. So all of these resources are being built on the same data and software platform, so I think it's going to be a really exciting way, and I'm really glad that the musculoskeletal data sets are going to be in at this phase as it grows. So I believe you can get to it. Here's the website right now. It'll be fully functional. We're still working on making it a little bit better, but there'll be the full launch will be coming very soon. And again, we've got data right now on bone mineral density, fracture risk, lumbar spine density, and osteoarthritis will be added soon. And what we want from you is your data. So, and we want you to use the site. So if you have GWAS data for another disease, please you can contact me later, and we would be very interested in putting it on our list to add to this site. So we hope you use it, and send suggestions if you have recommendations on how to improve it. So I'm going to turn the podium over to Dr. Ray, who's going to introduce the rest of the program.
Three speakers, and the format is that First, we will let our speakers present their work, and at the end, we will have a panel discussion with our speakers together. So, today, our first speaker is Dr. Shaolin Wu from Washington University, presenting on using single-cell RNA sequencing to demonstrate cellular heterogeneity and trajectories of lineage specifications.
Thank you very much for the introduction, and thank you for ORS for giving me the opportunity to present my workshop here. So this is my disclosure. First of all, I don't have a computer science or statistical degree, and I don't create toolkits for single-cell RNA sequencing analysis. However, what I'm really interested is to apply and modify current existing toolkits to study biological questions in musculoskeletal tissues. And other than this, I have nothing to disclose. So why do we care about single-cell RNA sequencing? Well, bulk RNA sequencing can give us so much information already. Because one of the problems for the bulk RNA sequencing is for the heterogeneous tissues, we actually pull all the RNA, or what we call cDNA, from the different cell types together. And when we perform the sequencing, it turns out the gene we observe here is actually averaged from all the cells. Well, for the single-cell RNA sequencing, one biggest advantage is for the heterogeneous tissues, you'll be able to barcode our mRNA, which I will discuss this in detail in a later slide. And after sequencing, we will be able to cluster our cellular populations. As you can see here, we may have a different ABC3 specific populations here. You can see that the gene expression could be very different among these populations. And one really interesting analogy to think about bulk RNA sequencing is like kind of drinking mixed smoothies, like it's a really rich smoothie, and you can taste all the different flavors, but you just don't know what they are. But when you do the single-cell RNA sequencing, it's like you're drinking a rainbow smoothie. You can see the clear layer here, and it will tell you a specific component within your smoothie. And hopefully you might be able to identify precious, component in your smoothie. So for the outline today, the overall aim is to basically share our experience in the single-cell RNA sequencing, and we're going to use a case study as a HIPSC chondrogenesis. So the method we're going to cover today is dissect heterocellular heterogeneity and determine differentiation trajectory, map gene regulatory networks, and construct heterocellular signaling models. So why do we care HIPSC chondrogenesis? We know HIPSC is as a human-induced pluripotent stem cells as a really promising cell source for cartilage regeneration because they have unlimited proliferation capacity, and they can also differentiate into chondrocyte. So we can use these cells to repair our cartilage or use them as an in vitro disease modeling. The problem is how to robustly differentiate HIPSCs into chondrocyte without any off-target cells remain really challenging. Recently, our lab established a protocol for HIPSC chondrogenesis via paroxysmal methadone substitution. So what we did is we fed our iPSCs a set of defined growth factors and small molecules daily, and drive them step by step into mesosomal lineage until they reach chondroprogenitor we call CP stage here. And then we lifted our cells from traditional pellet culture supplemented with TGF-beta3. So to validate our protocol, we use 4 unique iPSC lines. As you can see, all the lines show the really robust sepal staining with chondrocyte morphology. So this is actually really exciting and promising results for us. But every time when we look at our pellets, you can see our chondrocyte is pretty homogeneous within the pellets, but you still can see these off-target cells just sitting at the edge of our pellets. And we actually don't know what they are, but we do know they are not chondrocyte because they don't have chondrocyte morphology. So in order to figure out their identity, we decide to use single-cell RNA sequencing. So overall, we have 9 time points for single-cell RNA sequencing, and 3 of them from our mesodermal lineage stage, and 6 of them from our chondro pellet culture stage. So the platform we use for the single-cell capture is commercially available from the 10x Genomics. And the reason we pick up this platform because it's really efficient and fast, and most importantly, it has a low cost. So according to their website, to prepare the cells for this platform is really easy. So you can, when you harvest your cells, you can just freeze them in the regular freezing media and until you need it. So on the day of sequencing, you can thaw your cells, wash, and resuspend in the wash buffer. At for our case, we resuspend in the 2,000 cells per microliter. But this number may change, depends on your cell phenotype. And if you see a lot of dead cells, there's a kit, actually you can remove the dead cells. In terms of library preparation, in our case, we normally send our samples for the sequencing core to prep our library, which is more consistent. So this is just a brief introduction how single cells being captured. So for 10x chromium controller is actually a very simple microfluid device. Now you can see the fluid with the beads actually comes into one end, and the beads is conjugated with a barcode and the primers. They are actually small nucleotides. And the cells flow in from this end, and the oil comes from here. And the viscosity of the oil and the speed is adjusted in the way that when oil droplets forms, it only captures one single cell and one bead at a time. And the cells will then lyse within the oil droplets, and the reverse transcription is performed. And after that, the cDNA of our transcript actually conjugated the cell barcode, which come from our beads, and also conjugated a small nucleotide called UMI, which stands for Unique Molecular Identifier. And these UMIs actually we will use to count our genes, so we will be able to avoid bias from our PCR application. So the whole thing is amplified and sequenced and aligned to the human genome. In our case, we obtain around 4,000 genes per cell, 2,000 to 3,000 cells per sample. But again, this number may change, depends on the tissue you study. So in terms of animation here, you can actually see the red is our beads comes in, and the yellow is our cell. Now you can see this is one oil droplet here. Now you can see the beads is actually way bigger than our cells. But how do we know every time we only capture one single cell? To do this, we can actually verify by, we call this a mixed species experiment. So what we do is get some human cells and mouse cells, and we mix them together and perform single cell RNA sequencing. And if the oil droplets only capture one single cell at a time, if you capture mouse cell, it should be able to align back to mouse genome. Or capture human cell, the cell should be able to align to human genome. So whatever in between is actually when the oil droplets, it captures more than one cells. You can see the gray dot here, we call this multiplet. So you want this multiplet as low as possible. Normally you want it lower than 5%. And in our case, you can see it's 1.4%, so it's pretty good. And after sequencing, 10x Genomics actually provide a really useful pipeline called Cell Ranger. This Cell Ranger helps you to align your reads, and importantly, also generates, we call these gene barcode matrices. And this is a raw data you will actually obtain from your sequencing core. And we will use these to perform downstream analysis. So the first, I would like to talk about how we determine cellular heterogeneity by Seurat R package. So the gene barcode we obtained, the matrix, we will then input it into Seurat, which is R package developed by Satija from NYU. And the R is actually a programming language. It comes with a free editing environment. So then we removed a low quality of the cells that is less than 200 or more than 7,000 detected genes. And this number may change, depends on the cells you study. And we also remove genes that are detected less in 3 cells. So these steps, we call this quality control. And actually in their website, the instructions are very, very clear. You can just follow the instructions. And one important function embedded in the Seurat R package is actually called t-SNE. So t-SNE is an algorithm can perform nonlinear dimensionality reduction. And basically, because our gene barcode matches is really high dimensionality data, so t-SNE can help us reduce it into a 2D and visualize it. So now this plot we call a t-SNE plot. And each dot in the t-SNE plot is actually a single cell. And the cells will share the same color, represent they have a similar gene expression, so they tend to cluster together. And then you can see the cell cluster is actually our cellular population. You see this in this plot, we have 7 populations. And one of the advantages of the t-SNE in the Seurat is also give you enriched genes, a list of genes in a specific cell population or differentiated expressed genes. For example, in this brown population is enriched in SOX9 type 2 collagen. And with this gene, we can actually start to annotate our cellular population. So you actually look like this. And then I will also use this gene to perform gene ontology analysis. There are several websites or software you can use, but I prefer to use David gene functional classification tool, particularly looking at biological function. And then also if I compare the markers from current literature, now you can start to determine your cellular population. So now we know we have 3 populations belong to chondrocytes. They enrich for type 2 collagen, SOX9, and COL6, where we also have some neural cells and interestingly melanocytes in our chondrogenic pellets. Once we identify this heterocellular population, next, we're going to talk about how we determine differentiation trajectory, particularly by using a Monocle R package. So if you remember, we actually submitted 9 different time points for single-cell RNA sequencing. So we have 9 different sequencing data. So what we're going to do is we're going to take these data sets from different time points and then merge them into one single object in Seurat. And the merged matrix would then can be converted into Monocle object using the function called import CDS. And then the Monocle is actually developed by the Transcriptional at the University of Washington. And I highly recommend if you want to do this using a high performance computing core, because it's a very computing intensive process. So with Monocle, we can actually construct differentiation trajectory of our samples. Now in this principal component plot, each dot represents a single cell, and the cells you see that comes with colors. And the colors actually represent the time point of our cell. So now we know this HIPSC is my starting time point. So now you can see some cells start to gradually differentiate into chondrocyte. At some point, you see there's a lineage application, because that means some cells actually goes into neuron cells. And now that results into 2 branches. One is chondro branch, one's our neural branch. And one interesting function Monocle provide is actually you will be able to perform dynamic gene expression. So now if we look at chondrogenic markers, you can see these chondrogenic markers are enriched in our chondro branch, including aggregate COL2 and SOX9. When we look at the neurogenic markers such as nestin, OTX2, and Wnt3A, actually enriched in our neurogenic marker, neurogenic branch. So once we identify these off-target cells, our next step is actually going to reconstruct gene regular network we call GRNs. That is responsible for this off-target differentiation. So we're going to use an algorithm called WGCNA, which stands for Weighted Gene Co-expression Network Analysis. And this is developed by Dr. Horbers at UCLA. So the definition of the gene regular network is a collection of the genes with a similar expression patterns. And these genes may be responsible for regulating biological processes such as proliferation or differentiation. And the algorithm of WGCNA can actually help us identify clusters. And actually in this case, we call modules of highly correlated genes. And once we identify these modules, we can further identify hub genes, or we call driver genes, that is responsible for this differentiation. Again, we highly recommend using a high performance computing core to perform this process. So the overall aim for this WGCNA is trying to, in a big network, we're going to find a small network in which the genes are highly correlated with each other. So in a simplified example, assuming we have 6 genes here, A, B, C, and X, Y, Z, and we can actually calculate their correlation coefficient between genes. And this is based on, we're going to base on the assumption that co-regulated genes will have correlated expression. So once we calculate their coefficient, you can see that it seems like A, B, C genes are highly correlated among each other, while X, Y, Z are highly correlated among each other. And with this number, we can actually perform clustering and plot the clustering dendrogram. And you can actually using the algorithm we call random tree cut and make a cut here. Now you can see ABC genes actually highly clustered together. You can see they are belong to one module, or XYD belong to another module. And in WGCNA package, you can actually determine what is the height you want to determine to make your cut here. So you can change the module and the number of the modules you would like to see. So in the real deal, our data actually look like this. It's way more complicated, as you can think. So each is all different clusters. And when we make a cut here, now you see we identify 11 different modules. And each module is actually represented by a single color. So now what we can do is actually to extract genes from a specific module. For example, if we look at the green module, and what I did is I extracted the genes from this module and performed gene ontology analysis, as I mentioned before. Interestingly, we find that these genes are actually enriched for central nerve system, brain, and the forebrain development, suggesting that this module may be important for neurogenesis. And my next step is to, from the gene list, I start to identify transcription factors within my gene list. And then I identify top 100 genes that are highly associated, highest association connect to this specific transcription factor. Now you can start to imagine our network will probably look like this. But actually there's a really nice software, it's a free online website called CyTOF can help you visualize your gene regular network. So eventually our gene regular network will look like this. So let me briefly explain this. So each node here, as you can see, represents one gene. And the larger the node, that means the higher connectivity. There are more genes connect to it. And the line between two genes represent their correlation. So the thicker the line is, means they have higher correlation. And now if you see the genes becomes orange, that means it's a parameter called between the centrality. We can think about this gene, when it becomes orange, it actually sits in the center of our network. So this is important. Now we know Win4 is actually our drive genes in neurogenesis. Well, in addition to neurogenesis, we also identify another unique gene regular network is actually interesting, is monogenesis. And we find that Win2B is one of the hub genes. So now we see these two unique Win4 and Win2B ligands. And the question is, which one should I target? And another question is, seriously, how many different type of Win ligands is produced in my chondrogenic sample? So to answer this question, I start to use a dot plot function. You can find this in Seurat. So this plot, in each dot represents a specific gene. And the size of the dot represents the percentage of the cells express that gene. So now I can look at all the Win ligands from canonical and non-canonical Wins. So from this plot, you can appreciate that most Win ligands were actually produced by our off-target cells. So what does this mean? Does it matter for our chondrocyte? That means that can our chondrocyte receive actually these Win ligands? So to answer this question, I'm going to use Win3A as an example, as well as his high affinity receptor we call FRIZO2 here, because I'm to construct heterocellular signaling models. So in Seurat, I can actually construct a heat map here. In the heat map, each column represents a single cell. And the column with the red color means that that cell expresses specific gene. So now you can see for Win3A, most of Win3A was actually expressed by our neuron cells, not so much from our chondrocyte and the melanocyte. Where for FRIZO2, it actually expressed across our three major populations. And we can actually quantify the percentage of the cells express a specific gene. And with this information, by using a circularized R package, now I can build this heterocellular signaling model by ligand receptor interaction. So now this plot reads like this. So around 10% of the neuron cells produce Win3A, and 45% of the melanocytes can actually sense Win3A because they express FRIZO2. And Win3A can actually signal through a third of our chondrocyte because of receptor expression. And Win3A can also function as a paracrine or autocrine back to its neuron cells. So this is just one of the examples. We can actually look at all the Win ligands where the chondrocyte can sense Wins. Now, there are several several databases, you can find out ligand receptor partners. So all these databases are actually manually curated because every year you have new publications pop out, so they will actually update these. So with this information, we then ask the question, now, can we eliminate our target cells by targeting Wnt signaling? So we take out one of our iPSC line, and we treat our pellets with a Wnt inhibitor, which is a C59. Now you can see pellets treated with a Wnt inhibitor produce a much larger cartilage matrix and with reduced off-target cells. And if you recall, instead of Wnt, we actually have another potential top genes from monogenesis, that is MITF. So I actually find another inhibitor, small molecule inhibitor we call ML here. So I find that when pellets treated in combination with C59 and ML, it even further enhance our chondrogenesis. This is really, really nice pellets without any off-target cells. And when the pellets just treated with ML alone, you see it didn't work out at all. So this is just one of the examples. We can use single-cell RNA sequencing and associated bioinformatic analysis to identify target cells and they're responsible gene regular network and hub genes. And by targeting these molecular targets, we will be able to significantly enhance our iPSC differentiation. And this is actually an in vitro study. We can definitely use single-cell RNA sequencing in vivo. So these projects is actually led by our former postdoctor, Natalie Kelly. And actually the paper just published recently in Matrix Biology. So she's interested in investigating the cell phase specification during embryonic limb development. So what she did is she isolated findings from a different time point embryonic stage and performed single-cell RNA sequencing. Now you can see she will be able to identify all different cellular populations at different time points. And she can also evaluate their dynamic changes. And with WGCNA, she can also identify the hub genes that drive a specific lineage substitution. So in a quick summary, we use the red, we will be able to review cellular heterogeneity, and use Monocle, we can delineate differentiation trajectory. Well, WGCNA allowed us to construct gene regular network, and the red and the circularized can construct as heterocellular signaling models. And actually more detailed method, including functions I used to develop this process, actually described in handouts. And you can find it in our ORS 2020 workshop website. And with this, I would like to thank our funding source, our lab mates. And if you are interested in more detailed experiment results regarding how application of single-cell RNA sequencing on iPSC chondrogenesis, I will have another talk tomorrow. Please join us. Thank you very much.
So, good afternoon, everyone. First of all, I want to thank Jen and Farouk for organizing this workshop and hope you all are enjoying it as much as I have. I also want to thank the earlier speakers for very nicely introducing single-cell biology and omics. And since we have talked about RNA, DNA, and chromatin, so of course it's time to talk about proteomics. So I want to first just say that my disclosures are on the website and I have no conflict. I should have put that disclosure that I'm also not a computer scientist. I like that with Dr. Wu's presentation. So let's talk about mass cytometry, which is a single-cell proteomics technique which we can apply and we can map almost 40 probes at one go with this technique. And then by using some overlapping markers, you can actually even increase it to 80+ probes. So this is basically sort of, the idea is similar to flow cytometry of FACS, which we routinely use. But instead of fluorophores, here the antibodies are labeled with non-biological elemental isotopes, each of which has a unique mass. So instead of fluorescence, the readout here is a mass spectrometer. And the advantage there is that with the fluorophores, because of compensatory issues, you become limited to almost 18 to 20 probes. And here you can use many more probes, so the sample set becomes larger. So essentially, the cells are stained with antibodies, which are labeled with these elemental isotopes, as I just said, and then pass through a nebulizer, which basically converts them into single-cell droplets. And then these cells are passed through an inductively coupled plasma, which is at a very high temperature, so that basically the cells are vaporized, all the molecular bonds are broken, and now they are converted. Each of the single cells is converted into a mass of elemental ions, which can then be quantified by the mass spectrometer. So as a result, what you then have is basically, oops, okay. I'm going ahead myself. Sorry about that. It's trying to. So basically, what you have is you have a cell which is now quantified by all these elemental isotopes, so you can have a quantitative idea of how it has been labeled with these antibodies. And then this really high-dimensional data is then processed further, and what it can tell you is basically what are the expression and fold changes for each of these antibodies, as well as you can really figure out clusters of cells which are similar to each other, so you can look at distinct cell populations within this, within the amount of cells. Okay, so then this sort of approach, of course, is complementary to single-cell RNA-seq, because you can have a proteomic readout to the transcriptomics. Of course, it's not at a whole genome or a whole proteome level, because the number of probes is limited. So even though, so basically that is the limitation, that the probes have to be pre-selected. So in that sense, it's not really an unbiased approach. But as I said earlier, you can use 40 plus or even 80 probes as compared to FACS. Another advantage is that you can actually look at active signaling pathways, which is something you cannot do with the RNA-seq. And millions of cells can be analyzed in a cost-effective manner. So especially if you're looking for rare populations, which become important when you're looking at patient samples, then that sort of an analysis is very difficult or becomes very cost prohibitive to do with RNA-seq, where typically it is possible to sequence only a few thousands of cells. But whereas with CyTOF, you can actually look at millions of cells. So specifically, if you're interested in rare cell populations, mass cytometry is a good approach. So this technique was developed by Sean Bendel and Gary Nolan now almost a decade ago, and it was developed really for the blood and for looking at the immune cells. And since then, the immune signatures have actually been looked at in many diseases, including blood as well as synovial tissues in rheumatoid arthritis, as well as these single-cell immune signatures were looked at before and after a hip replacement surgery to see which signatures can actually predict the surgery outcome. So my lab is very interested in osteoarthritis, as I guess most of you here would agree. It's an unmet clinical need for which there's no disease-modifying drug treatments available. And the early events in OA pathogenesis are still elusive, and the economic burden of this disease exceeds almost 50 billion USD. So I thought today I want to share with you some of the recent data from the lab. And with that, I'll walk you step by step through a mass cytometry based approach. So osteoarthritis, as we know, is a disease of the joint. Basically, the end-stage disease is characterized by cartilage degradation, bone remodeling, as well as an inflamed synovium. So really, the whole joint environment is altered. And we know that cartilage or joint injuries greatly increase the incidence of OA. So we thought we'll start with a study of the cartilage per se. At some point of time, really, we would like to reconstruct a single-cell atlas of the whole joint, but we started these studies with a focus on cartilage. Now, cartilage, as again, many of you would know, is a very interesting tissue in that it does not seem to regenerate very well. As compared to bone, if there is a cartilage injury, even when you are a healthy 16 or 18 years old, really it does not translate into full recovery of the injury and actually increases your incidence or your risk of osteoarthritis. So the reason for that, most of the time, of course, there's a low regeneration potential, but why there's a low regeneration potential, some of the reasons which have been described are that there's a low cellularity. Really is a tissue with just chondrocyte embedded within its ECM, and the cellularity for the tissue is low. It's avascular, that is, there is no blood supply, so there are no stem cells coming in from the blood supply, and probably that is the reason why the regeneration is low. However, there have been many studies where putative progenitor cells, cartilage-resident progenitor cells, have been identified and described. So really, we wanted to use these single cell studies to try and understand what are these cartilage progenitor cells and why do they basically not do a job very effectively. We also wanted to look at some disease-associated populations in the hope of finding some targets for disease-modifying drugs one day. So this is basically the layout of our studies, and this is something which will come online next month. I think it's in Press in Science Advances. So what we have done is we profiled 6 normal chondrocytes, which were procured from commercial sources, and 20 OA cartilage. So this cartilage has been taken from patients undergoing total knee replacement. So really, it is an end-stage disease cartilage that we're looking at. And as I said before, you derive these cells. We usually just expand them very minimally in a high-density monolayer for a day or two. And then the cells are stained with this 33 marker panel, which I'll describe to you in a second. The cells are then nebulized, and the mass cytometry data is collected. Then, just to make sure that we're looking only at cartilage cells, we gate out for markers for chondrocytes, that is CD44 and SOX9. And we also use cisplatin to basically discard any dead cells, so that we're looking only at live cells. And then we go ahead and do the data analysis. So the panel that we have used here, we've actually used a lot of cell surface markers, which are basically putative cartilage progenitor cell markers. So there's a list of those. CD44, which is a chondrocyte marker. There are cell cycle markers, a lot of signaling pathways, and some inflammatory receptors. So as you can imagine, when you do 26 samples with 33 markers and looking at 1 to 2 million cells per sample, that really leaves you with around 250 million data points. And the question is then, how should you analyze them? So I'll run you through 2 common algorithms which are used to analyze this mass cytometry data. One of them is FlowSOM. So FlowSOM basically uses the idea of self-organizing maps for the clustering as well as visualization of cytometry data. So in this case, the FCS files are read, they are compensated, and then scaled, and then basically generate a matrix where each cell is associated with a matrix of the parameters, which is basically the 33 marker parameters. And then the algorithm starts to generate these self-organizing maps of cells, which are very similar in terms of this matrix. And for visualization, then these clusters or nodes are actually connected together in what is called a minimum spanning tree, which basically has a least weight associated with this. And that is how you can visualize this data. So this is just one way of visualizing the data. So when we run a FlowSOM algorithm on our data, we basically get a minimum spanning tree representation for normal and OA samples, as I show you here. So here, this node is a cluster of cells which are very similar to each other, and the algorithm has recognized it as a single cluster. You can see the same cluster in the OA samples as well, as represented here. So another way to visualize this high-dimensional data is what was shown by Dr. Wu as well, and that is t-SNE. So t-SNE is basically T-stochastic neighbor embedding is the full form, but the idea really is to try and depict data which is in 3-dimensional in a 2-dimensional format, but maintaining the spatial distribution of the data. So for example, you see here in the 3D data, these blue and red cells are really far apart, and that spatial distribution is maintained in this 2D plot as well. So when we do a t-SNE representation of our data, you can see clearly here that the OA samples and the normal samples actually have a very different spatial distribution. All the averaged OA samples are in red here, and the normal samples are in blue here. And you can see that they are quite distinct because they occupy very different space. When we look at single markers, we sort of see that they are all pretty high for CD44, SOX9. But when you look at Notch1, you see that in the normal cells, it's very low expression, whereas in the OA cells, there is expression, but it's quite varied. Some clusters are high in Notch expression, while others are low. And NF-kappa B has actually a quite broad distribution. So another algorithm which is also used frequently in the field is the X-shift algorithm. And here, basically, the algorithm provides a K nearest neighbor density estimate, and it goes through different K numbers, basically to try and model the data best. So what it does here is that it provides a density estimate to each of the neighboring cells, and then it tries to look for a local maxima and kind of organizes the clusters around it. So you see that here, basically, this then becomes 3 clusters, and it keeps on doing it till it sort of reaches what is called an elbow point, where adding more clusters does not really model the data any better. So basically, it is trying to have a minimum number of clusters which would explain the data quite well. So we did this as well for our data, and this provided us with 24 clusters. From FlowSOM, we get 20 clusters, and the nature of the clusters is actually quite same. So this was just to validate that, you know, we were generating the right amount of clusters to explain this data by FlowSOM. So basically, that means that in our chondrocyte data, we have 20 clusters or 20 distinct cell populations in the normal as well as OA cartilage. So now let's think about what are these clusters and what is their molecular identity and what do they do. So this here just presents a heat map of all the different markers that we have used in the panel. And these are the different clusters. So you can see clearly that each of the cluster is quite distinct, right? If you look at the heat map, they are their own special snowflake. And when we look at the distribution of these clusters in normal and OA, one thing which becomes very clear is that most of these clusters are present in both normal and OA. I'll talk to you about things which are depleted or enriched in just a moment. But we clearly see that the distribution is much more variable in the OA samples. So that would mean that the OA samples or the OA patients are much more heterogeneous in the distribution of these clusters. And we see the same thing when we actually calculate a coefficient of variation, that these clusters, they show much less variation in the normal samples as compared to the OA samples. We also did this in a separate way. This is Shannon Diversity Index, is something which is more of an ecological concept to see how different 2 cell populations are. And here also we see that the OA population is much more heterogeneous. When we think about the molecular identity of these subpopulations, as I told you, our panel is very vested towards cartilage progenitor markers. These are different markers which have been described for these CPCs. These CPCs are supposed to be migratory in OA, and they do give rise to chondrocytes in culture. So the question really is, why do they not work in vivo? And when we look at these subsets, we see that a lot of them are CPC subsets, but then there are some non-CPC subsets. So we found around 7 non-CPC subpopulations, and the rest of them are CPC populations. But then we see that CPCs come in many flavors. There are CPCs which are enriched in OA. There are others which are depleted in OA, and some which are similar. So of course, then we are very interested in these CPCs which are depleted in OA, because maybe these are the regenerative, proliferative populations which are lost during disease. When we do just unbiased, unsupervised clustering, we can see that there are, actually, sorry, this is not unsupervised. This is basically just CPCs. We just divided them in 3 parts based on whether they are depleted in OA, whether they are similar in OA, or whether they are enriched in OA. And you can see that the CPC 2 is the one which is similar between normal and OA, but CPC 1 are the ones which are depleted. And when we look at the cell cycle characteristics of these CPCs, it is interesting that most of the CPCs which are depleted in OA are actually the one which seem to be more in the cell cycle. So of course, these are chondrocytes, so these are very long, they don't divide very fast, and that is sort of reflected here as well. But we can see that the CPCs which are present only in the normal cartilage actually seem to be more proliferative than others. And then we also see that the signaling, if you look at the signaling pathways, it seems like CPC3, which is enriched in OA, is actually quite inflamed. So 2 of them are not so inflamed, whereas 3 of these CPCs are actually very inflamed. So this is interesting as well, because maybe a high inflammation could actually poison these CPCs, so they don't work so well. And that is just a hypothesis that we have to test, but it's interesting that there is a variation in these CPCs in terms of inflammation. CPC2, which is present in both OA and normal, are actually quite low in inflammation. Then we wanted to sort of really ask this question of whether now knowing all these cellular populations can help us figure out what is different in these 20 OA patients. And of course, it's a very small subset, but here, when we did an unsupervised hierarchical clustering, we can sort of see that there are clearly 3 different groups of OA patients, A, B, and C, and the normal cells also segregate very nicely out, so it shows that that's a different subset here as well. So this sort of a stratification of the patients is interesting. So this is like, again, just a colorful Lego representation of the different groups. But here what you can sort of see is that these are the different subpopulations that we have identified. And you can clearly see that, say, subset 8 is very highly represented in the normal cells or in the normal healthy adults, but it's not really apparent in the patients. And there are these basically represent the frequencies of these clusters in these 3 different groups of patients. And they are clearly very different in the kind of CPCs they are enriched in as well. So when we look at the inflamed CPCs, which are the Notch 1 positive CPCs, you can see that group C actually has more of these CPC clusters. But when we look at CD105 plus CPCs, they are more in groups A and B. So this is sort of an interesting idea that maybe different patients would respond differently to certain drugs which can actually cause a regenerative environment, or drugs which actually dampen inflammation based on this sort of a distribution of the cell clusters. So then the next we wanted to look at some of these non-CPC populations, and what we found was that these non-CPC populations were actually very rare. So these are cells that we're talking about, just 1 to 2% of the whole population, but they were very consistently there in all the OA patients. And now we know from the work of Jennifer Ellisef and Judy Campisi that rare populations can drive pathology, including OA pathology, because in this paper, when they targeted senescent cells, which make, again, 1 to 2% of the whole cartilage, upon the abolishing these senescent cells with senolytics, what they saw was that OA was impaired. So here you can sort of see from this paper that this is no surgery. This is when you have ACLT with vehicle, and when the senolytic is used, which basically gets rid of these senescent cell population marked by HMGB1 and P16, there was cartilage degradation was significantly impaired. So we know that rare populations are important, and with that in mind, we set forth to think about what do these rare populations mean. So there were 2 clusters which we noticed were actually quite distinct from the rest of the clusters, and these were clusters 15 and 20, which were marked by TNF-R2 and IL-1R1. So they were double positive for these 2 inflammatory markers. And so while cluster 15 is present in normal cells as well, cluster 20 was actually present only in the OA cells, and it was significantly much more enriched in the OA cells as compared to normal cells. We also noticed, then we took advantage of RNA-seq dataset, which had been published in 2019, And from that, we pulled out these double positive cells and looked at their transcriptome. So this is a nice way to sort of show how the RNA-seq, single-set RNA-seq data can complement the proteomic data. And what we saw was that these cells basically were enriched in immune response and in immune system, inflammatory response. And based on this, we thought that this was actually inflammation associated or an inflammation amplifying population. Then we also noticed that this population signaled specifically through JUNC and SMAD1/3. And this was only these clusters which signaled through this. None of the other clusters actually were high for these populations. So then this, of course, begged the question, can we now modulate this Inf-A population, and what would happen? So then when we use a JUNC inhibitor in 6 additional OA patients, they are not from the original cohort, this is a different validation cohort, lo and behold, when we looked at a 62 cytokine Luminix panel, what we saw was that clearly CCL2 and CCL7, the monocyte recruiting cytokines, were greatly reduced in these OA cells upon treatment with the JUNC inhibitor. And this was impressive because the loss or inhibition of a population that is only 2% of the total population reduced the secretion of these cytokines by around 50%. And when we used an NF-kappa-B inhibitor, we did not see the same thing. So this is the middle panel where we used an NF-kappa-B inhibitor. This is the JUNC inhibitor data. And then we went ahead and inhibited ALK, which is upstream of SMAD1/3. And we saw the same result, that CCL2 and CCL7 were downregulated, and CXCL1 and CXCL5 were further downregulated. So clearly, inhibition of Inf-A seems to be beneficial. And here I just want to remind you that the CCL2 negative mice are actually protective from OA. Also the CCR2 knockout mice, CCR2 is the receptor for both CCL2 and CCL7, and they are also protected from OA in this mouse model of surgery-induced OA. So this means that these two cytokines are actually quite important. So then we went back and looked at one of our favorite markers, CD24. So this is a marker which we had identified a couple of years ago when we did a transcriptional profiling of juvenile versus adult cartilage. And we found that this marker was enriched in juvenile cartilage. We also found that this marker was enriched in iPSC-derived chondrocytes. So CD24 is a small glycosylated cell-anchored protein, and it is known to actually regulate NF-kappa-B. And what we found was that when we knock down CD24, chondrocytes do not respond as strongly to inflammatory cues. So the presence of CD24 actually protects the chondrocytes against inflammation. So that's why we actually went ahead and looked for this marker in our data. And we found 2 clusters which were positive for CD24. And as we had seen before, we saw that this cluster, even in normal chondrocytes, gets lost with age or gets reduced with age. And when we pulled out the transcriptional profile from the single-cell InAC data, again, we found inflammatory response and immune cell trafficking as being one of the major pathways which are up. Another interesting thing we noticed was that we could divide all the OA patients into Inf-D low or Inf-D high. This is the inflammation dampening population. And this Inf-D population was actually very nicely correlated with the Inf-A population that I just described before. So basically, if the patients had low Inf-D, they had low Inf-A. If they had high Inf-D, they had high Inf-A. Of course, this is just a correlation, but from this we got the hypothesis that what if we now modulate both the populations at the same time? That is, we inhibit Inf-A and activate Inf-D. And sure enough, when we do that, we saw, so this is just an activator of CD24, which does not have any significant effect. But when we activate CD24 and inhibit JUNC, we see a bigger effect than JUNC alone. Okay, so then what is coming out from this analysis is that paracrine signaling is very important. And there is a crosstalk between these inflammation amplifying and dampening populations, which really regulates the secretome or the microenvironment of the OA cartilage. So in summary, single-cell proteomic studies therefore have a great potential to provide a deeper understanding of healthy and diseased cartilage or skeletal tissues. And this can allow stratification of patients and thereby lead to a better precision medicine sort of approach, which I think is really needed for OA patients if we are to have a meaningful therapy at some point of time. With this, I want to thank my lab. Most of the work here was done by Fiorella Grandi, a graduate student in the lab, my collaborators, funding sources, and thank you very much for listening.
We would like to the presenters to come to the podium, please, and we will take a couple of questions.
It's a good question. I'm not a geneticist either during the day, but these are all published results. So we're taking them from, we're assuming that these issues are dealt with at the review stage. So I'm sorry, I can't really answer that question. Yeah, I'm sorry, I don't know the answer. What's that? Can you repeat that question? It was a little hard to hear up here. Please.
So you construct basically, and you do it again. The island positions they change. So how do you compare two different universes? And A, you have or opposite are together. So that's what I have. And I will not. Yeah, I thought it was really important. So actually, instead of the principal components is the linear reduction. We actually observe the distance with different components. But this is, it does not answer the global structure as well. It's more like so I read the paper before when you look at the single cell data species, it's the same class, cell class, the distance between two cells conserve the overall structure of the cake. But you manage time healing, so other people find it very
Last question, microphone number 2. Who is the question for? Dr. Wu? Is dead cells, removing dead cells? Yeah.
So for my simple, because I practice my method, I don't really see something. So probably to use some of the like about 60 to 80%, that might be. Why don't you do this? The probably if you do like mouse easier, it's more harsh. You might use using the I can also comment on the taxes. We generally try to sequence these tax cells where there's less than 5% of cells that the cells that reflect. Now, what we were doing here was collecting population cells from different parts of the body. We tried to do this from growth frozen stem cells. And we had a very hard time with live cells because there's only frozen, and then we had to extract cells. So we did a lot of observations on media, transport media, or transplants, and be able to get viable cells and extract the cells and do experiments on them. And one thing we had to optimize before we did any of this is whether or not we were changing the genome in response to that media before we were out. We performed similar analyses on mouse embryos and treated, ship cells and treated exactly what it was. And we did the exact protocols. We found about 90% overlap of our insects between fresh shipped and fresh collected, which is probably what's actually do this. The other thing that we did is kind of really one of the practices that we built that out is we use the collagen tool cleanouts to fax for GFP chondrocyte populations from what we just go ahead, and then we do taxes on those populations, comparing those to the second population. We got about 85% overlap of all the insects that we looked at. So we were pretty sure we were collecting population cells that are really positive. The reason why we do that is because obviously there's not good facts out there for collagen chondrocytes in insects. You have to know where you're playing with the mouse before you know what you're doing. So there's a lot of troubleshooting steps that we did that we not doing labs, and it was interesting doing these things that were on in vivo tissues like mouse and moose samples. These reach out to me on lab and all the different steps we did, including analyzing work and so forth. Often in the taxes, go to the literature, go online, forums, and black box. It's very hard to do. Please reach out to me. My email website.
Great. Thank you. Let's thank all the speakers one more time, and we'll get you onto the study section, onto the poster session. If you have extra questions, the speakers will stick around for the last. Thank you. Oh, okay. Come down here. Where do you want to go? Just right here by the door.